So, good morning to all of you. Then, in the last class, we have seen the classification of five kingdom classification by Whittaker. He divided the living organisms into five kingdoms based on 3 points: complexity of the cell structure, whether eukaryotic or prokaryotic; complexity of the body structure, whether unicellular or multicellular; type of nutrition, whether autotrophic or heterotrophic. Based on that, Whittaker in 1969 divided the complete living organisms into how many kingdoms? 5 kingdoms. That classification is called a 5 kingdom classification, and that kingdom include 5 kingdoms. First of all, kingdom Monera, Protista, Plantae, Fungus and Animalia. 5 kingdoms are there. In the coming topics, we have to discuss the detailed characters of each 5 kingdoms separately. And today we can start the discussion of first kingdom, that is called as kingdom first one. That first kingdom is called kingdom Monera. See, kingdom Monera. This is the first kingdom we have to explain in detail. So the first kingdom comes kingdom Monera. Which kind of organisms are coming under that? All the organisms under the kingdom are prokaryotic. All organisms under the kingdom are prokaryotic. What is the meaning of prokaryotic? Cells without nucleus and clear membrane bound structures are called, old type of cells are called prokaryotic cells. So all the organisms coming under the kingdom Monera are prokaryotic organisms or prokaryotic cells. And remember that all the organisms coming under the group are bacteria. All are bacteria. All bacteria comes under the kingdom Monera. So we have to discuss the characters of kingdom Monera, or what are the characters of bacteria in detail. Which type of bacteria are there? What are the classification of bacteria? And the most important character is that you know that they are without nucleus and membrane bound organelles. Nucleus is completely absent. So in such a group of organisms, in prokaryotic organism, clear nucleus is absent. Membrane bound organelles, what are they? Nucleus, mitochondria, chloroplast, Golgi bodies, etc. are completely absent. So these organelles are called membrane bound organelles. Membrane covered organelles, completely absent in the prokaryotic cell. Clear nucleus is absent. Somewhat nucleus is present, but nucleus is not clear. Then another importance is that the cell wall is very thick. Very thick cell wall is present. You know, cell is covered by a cell wall. Then we can discuss later on the structure of bacteria. The thick cell wall is present. Cell is covered by covering. That is called cell wall. So bacteria, cell wall is very thick. Thick cell wall is present in bacteria. Why the cell wall is very thick? Because bacteria are the first living organisms of the world. They originated crores of years ago, before plants and animals. At that time, the temperature of Earth was more than 100 degrees Celsius. You can imagine that. Plants and animals cannot live at that time. Bacteria lived at that time, crores of years ago, when the atmosphere was very hot. So the cell has to, organism has to protect. Bacteria have to get protection from temperature, high temperature. Because inside the bacterial cell, it is a living organism. So inside there is a chromosome is there, DNA is there. So to protect the DNA, cell have very thick cell wall. So all the organisms coming under the kingdom Monera are prokaryotic. All are bacteria. All are without nucleus and membrane bound organelles, and thick cell wall is present to protect the cell from unfavorable condition. These are the important characters of kingdom Monera. Then we can see the classification of bacteria. How the bacteria were classified? Bacteria were classified based on different types of bacteria are available. First of all, based on the shape. See, based on the shape, bacteria were classified into 4 categories. Based on the shape of bacteria that you studied in your school syllabus. Some of the bacteria are called coccus bacteria. Round shaped bacteria are called coccus bacteria. Then second type is called bacillus bacteria. Bacillus bacteria are rod-shaped bacteria are called bacillus. Rod-shaped, R-O-D. Rod-shaped bacteria are called bacillus bacteria. Third one, Vibrio bacteria. Vibrio bacteria is called Vibrio or otherwise called as comma-shaped bacteria. C-O-M-M-A. Comma-shaped bacteria like this. Bacteria having this shape are called comma-shaped bacteria or Vibrio bacteria. Then fourth one, some of the bacteria are called Spirillum. S-P-R-I-L-L-U-M. Spirillum bacteria. S-P-I-R-I-L-L-U-M. Spirillum means spring-shaped bacteria are called Spirillum bacteria. Spring-shaped bacteria are called Spirillum bacteria. So, based on the shape, bacteria, this is an old classification. Anyhow, different types of bacteria are available. So, bacteria were classified. You know what is the meaning of classification. Grouping is called classification. We are discussing the biological classification. Bacteria is a living organism. So, bacteria classified variously by different scientists. Different types of classifications are there. Anyhow, bacteria is classified based on the shape into 4 categories: coccus, bacillus, vibrio and spirillum. Round-shaped bacteria are called coccus. Rod-shaped bacteria are called bacillus. Comma-shaped bacteria are called vibrio. Spring-shaped bacteria are called spirillum. So, bacteria is based on the shape classified into 4 categories. Then another classification based on the staining property. Are you clear this one? Coccus, bacillus, vibrio and spirillum bacteria. Then based on the staining property. Remember this is important. Based on the staining property. S-T-A-I-N-I-N-G. Staining property. P-R-O-P-E-R-T-Y. Based on the staining property. What do you mean by staining property? Stain, S-T-A-I-N. Stain means some chemical substances giving color. It's called stain. To the cloth we are giving color. Like that we are giving color. That color is called stain, S-T-A-I-N. Staining property. Color absorbing property. Bacteria were classified into 2 types. Gram positive, gram positive and gram negative. Now bacteria were classified like the 2 group. First is called gram positive and second is called gram negative. What do you mean by staining property? Bacteria, what is the color of bacteria? Are you seeing the bacteria? What is the color of bacteria? We cannot see bacteria with our naked eye. We require microscope. And by microscope we can see bacteria. From where we get the bacteria? See, from where we get the samples of bacteria. Suppose you are asked to bring bacteria, 5 grams of bacteria tomorrow. How can you bring bacteria? From where we get the bacteria? Are you seeing bacteria? Are you seeing bacteria? Yes, bacteria. From where we can get the bacteria? Can you give example? Can you bring bacteria? Can you give example for bacteria for me? Yes, bacteria are available. Where it is available? See, very simple answer. You know, the bacteria called lactobacillus. Have you heard about the bacteria called lactobacillus? Where it is present? It is present in the curd. The milk is converted into curd by the bacteria called lactobacillus. The shape is bacillus. Lactobacillus, lactic acid, lactobacillus. So, bacteria is present in the curd. We can get the sample curd very easily. The curd contains bacteria. More than that, you know root nodules, leguminous plants. The root nodule, you can study later on. The leguminous plant, very example, you know the plant called touch-me-not plant. Touch-me-not plant. If you are plucking the plant, root contains small white swellings. See, touch-me-not plant or pea plant or beans plant. If you are plucking the plant, the seed, the root contains small swellings. White colored one. that swellings are called root nodules. In that root nodule, there is a bacteria. You know can you name the bacteria? Rhizobium bacteria living inside the root nodule. If you are taking the root nodule and just pressing like that, bacteria will come out of that. So we can get bacteria from curd, lactobacillus bacteria. We can get bacteria from root nodule, rhizobium bacteria. Like that, we can get samples of bacteria very easily. What is the color of bacteria? What is the color of bacteria? Bacteria are usually colorless. There is no color to bacteria. So we have to give color. Otherwise we cannot see under the microscope. So we will give color to bacteria. What color we are giving? We are giving a ink-like blue color called as crystal violet. See, the color is called crystal violet. See, crystal violet. See, a violet color. We are giving a violet color, ink-like color to bacteria. That color is called what? Crystal violet. So, we are taking in a test tube. We cannot do like that. Just I am doing like that. We cannot put the bacteria in the test tube. Method is there. Simply I am saying, and this is not explained in the textbook. So, we are putting the bacteria in which crystal violet. We are putting the bacteria in for the bacteria for 5 minutes in crystal violet. Putting the bacteria in crystal violet for 5 minutes. Then after, we are putting the bacteria in another solution called potassium iodide solution. We put the bacteria after 5 minutes in potassium iodide solution. So what is the color of bacteria? Violet color. So the bacteria is put in crystal violet for 5 minutes. The bacteria become which color? Violet color. Then after, we are taking the bacteria and putting potassium iodide solution. Potassium iodide solution for 5 minutes. Not 5 minutes, I am saying time, 5 minutes. Then after, we are putting the bacteria in another solution containing ethyl alcohol. See, ethyl alcohol. After that, putting the bacteria in ethyl alcohol solution. Ethyl alcohol solution. So we require 3 substances. We require. One is the one substance is called chemical is crystal violet. Putting crystal violet in 5 bacteria in crystal violet for 5 minutes. Bacteria become violet in color. Putting in potassium iodide for another 5 minutes. The bacteria is again which color? Same color, no change in color, same violet color. But we are putting the bacteria in ethyl alcohol solution for 5 minutes. Some of the bacteria will lose the color. Some of the bacteria lose the color, colorless. Some other bacteria is always violet in color. See, these 2 bacteria lose the color. It will become again colorless. This bacteria always violet in color. So remember that bacteria put in crystal violet, then put in potassium iodide, same color, no change in color. But when the bacteria is put in ethyl alcohol, what happens? Some of the bacteria will lose the color, violet color, so become colorless. But some bacteria always violet in color. If you are putting 1 hour or 2 hours, the color will not change. This bacteria which lose the color is called as gram negative, and which bacteria always violet in color is called a gram positive bacteria. The method was explained by a scientist called Gram. The name of the scientist is called Gram. The procedure was, the method was explained by the scientist called Gram. So the process is called Gram staining. This method is called Gram staining. Staining means the process of giving color is, stain is called staining. The process of giving stain color is called staining. So there are 2 types of bacteria based on the color absorbing property. gram-positive and gram-negative bacteria. You remember that the bacteria which is always violet in color is called gram-positive, which is lose the color, which become colorless when put in ethyl alcohol is called a gram-negative bacteria. So there are two types of bacteria, gram-positive and gram-negative. So this is the classification of bacteria. So bacteria were classified two types. Bacteria based on the shape classified into Coccus, Bacillus, Spirillum and Vibrio. That's an old classification. Now the bacteria were classified not like that. We classify the bacteria based on which color absorbing property, staining property. Bacteria are usually colorless. When they are put in crystal violet, bacteria become violet in color. Then put in potassium iodide for another 5 minutes. No change in color, same violet color. But when put in ethyl alcohol, some of the bacteria will lose the color, again become colorless. But some bacteria is always violet in color. Such bacteria which lose the color is called gram-negative, which is always violet is called a gram-positive bacteria. This method was explained by a scientist called Gram. So called what? Gram staining, gram-positive. To remember the name of the scientist, gram-positive and gram-negative. Why the bacteria become colorless? Why the bacteria always color? That we can discuss later on. That is based on the nature of the cell wall, nature of the covering of the bacteria. That may differ. Gram-positive and gram-negative is different. So the color loses or color retains. So this is about the classification of bacteria, and the more things we can see in the next class. I will stop the class now. I will send the notes for you today itself. So you listen very carefully and write the notes. Then we can see the more characters. Bacteria were classified into 3 categories. The characters of bacteria will come in the next class. Thank you.